Okay, next up we have a presentation on the Matchmaker Exchange demonstration project, and Kim Boycott is a Canadian local from the other side in the Children's Hospital of Eastern Ontario. But I must say my first glimpse of Kim from afar was in her leadership of both the Rare Disease Genes Discovery Project, or FORGE Canada, which has been incredibly successful, as well as a major leadership role in the International Rare Disease Research Consortium. So Kim has a great deal of experience in working in major collaborations.
So thanks, everyone. And I'm just going to take the next 12 minutes or so, I think, to talk on behalf of the Matchmaker Exchange Working Group in terms of what we're trying to do in this area of rare disease. And so the challenge that we face and have all come to realize is that there are 7,000 rare diseases, and for sure there are thousands more than that. Ada will nod. She doesn't like it when we say there are only 7,000. But we've had disruptive technology now for 4 to 5 years, and we all realize that we can't solve them all with it. So there was great promise at the time of whole exome and genome sequencing that we were going to sort all this out. Not quite the case. Now, there are a number of reasons that we don't solve a particular patient or family, and they're listed there on that iceberg, and why we can't sort of rise that one to the top. But what the community began to realize several years ago is it's the N of one, the single patient, that there's a place that we could possibly make a transformative impact. And so this represents the Lonely Exome, and this is my patient Noah. And he had his exome done as part of one of our research projects along with his parents who were unaffected several years ago. And he has remained unsolved. Now, Noah has cerebellar ataxia. He has a very good, compelling de novo candidate variant in a gene where if you knock that thing out in the mouse, it gives you cerebellar ataxia. But he remains alone, and therefore we do not have enough evidence to say that that gene is causative for his condition. And there are thousands and thousands of children like Noah. And right now, 60 to 70% of all exomes in rare disease are lonely. So this is the N of one problem, and what we needed to address is we need to find me over there in Canada, in Ottawa, with a set of phenotypic data on a patient like Noah, on his genotypic data from his exome, and I need to be able to find somewhere else in the world where I can find the same disease and then therefore be able to solve Noah's clinical issues. Now, this is how we've done it in the past. And this is actually a dinner from 2 years ago in Rome where I was a speaker at a workshop on dysmorphology. And this is literally where we solved 2 of our genes, diseases. And so that's Burnt Woolnough with ANO6 and Bill Dobbins with FGFR1, causing a very rare condition, even though it's a gene that's known to cause other things. But you can see from the number of wine glasses there that that is not a scalable approach. My liver enzymes would be through the roof. Now, this is 11 o'clock at night, and all they do is seem to drink wine after wine after wine. And so we got a lot of work done, and we made 2 novel discoveries that night, but we can't continue to do this. And so what I actually need to do is, with less alcohol, be able to meet Eamon Sheridan, who's another one where we have run into each other, through a genomic matchmaker. And essentially, that is a place that's almost a dating service for these lonely exomes, where we can describe a patient and the candidate gene that we think is important and be able to find somebody else who's looking on the other side with the exact same set of criteria. Now, many people thought of this problem at the same time. And as you can tell, guess what? We all stood up different solutions for the same issue of the N of one. And so in 2013, at this meeting when it was in Boston, Heidi and I and others, Ada and a number of people got together. We had no money for that meeting. Heidi went to the grocery store and got some juice boxes and those muffins. And we sat around the table and we said, these are all the solutions that we have stood up in various places in various countries for various consortia. And guess what? We all siloed our data, and that data is not talking to each other. So yes, this is great that we've all come up with a way to do this, but we need to actually figure out how to get to talk to one another. And so this launched the Matchmaker Exchange, which was a way for these siloed databases to actually speak to one another. So 3 years ago, I had never heard of an API. I had to look it up on Wikipedia. I didn't know what that was. Even Orion now, I'm not sure I know what it is. But we needed a way to have these databases talk to each other and exchange data so that as a clinician, with hundreds of unsolved patients, I don't have to put my data in every database. And so we developed the Matchmaker Exchange. It is an API. And we decided to go with a federated network so that each of these databases could talk to the other databases it wanted to and exchange data such that discoveries could be made without depositing in multiple places. Now, these are the 7 databases that are currently connected via the API. They are point-to-point connections, which means you exchange one-to-one, and this is where we've come sort of 3 years later. Now, I'll show you how this works, okay? So this is the story of how Dave meets Isabelle. Dave Diamond is a colleague of mine. One of his patients was studied in one of our large projects. And we collect all of our data in PhenomeCentral, which is a database that's located in Canada but is international. So Dave enters his patient who's got a nice-looking Candida gene. So there's a pedigree that goes in there. The next thing he enters is the clinical symptoms using human phenotype ontology, so it's standard computer-readable description of the patient. You can see there on the right. So there's growth parameters. This child's primary presentation is intellectual disability and epilepsy. He then flags the candidate gene, which is TRIC1. It's recessive. There are 2 mutations. And then we look within PhenomeCentral to see whether or not there's any other patients with that phenotype and that gene that look compelling. And you can see there down the middle, the relevance score doesn't look very good. There's no good matches in there. So the next thing you can do then is go to the Matchmaker Exchange. It's kind of like when you book a flight and you want to check different places. So you can go to Decipher in Gene Matcher, and in Gene Matcher, we can then see that there's a very high relevance score with a patient that Isabelle Trifot, who turns out to be Canadian, by the way, but working in Kansas City, have an important match. And so Dave and Isabelle met and have compared data and have now collected a cohort of 6 children who have compound heterozygous mutations in this particular gene associated with epilepsy, and that paper is about to be submitted, and you can tell it takes a year to get all that done and the functional work that goes with that. But this is a novel discovery that was facilitated through the Matchmaker Exchange. And so last year, just if you want more information, we did have a special issue that came out in conjunction with this meeting in Human Mutation. Our website is there. In that special issue of Human Mutation, we have a summary overview of what the vision is. We have details for those interested about the API, and then we have a number of examples from each of the databases as well as some successes. So we now have to look at this and say, okay, so we've come along this far, 3 years, where are we actually need to go next? And there are a number of places we need to go. I won't stand up here and tell you that we've solved all this, because we have not. And the first thing we need to do is increase the specificity of a match. And so you can look at that graph there and you can see phenotype along the bottom and the quality of the variant along the other axis. And really, where this will be most successful is in that sweet spot that I have circled there, where you have very good, high quality and discriminating phenotype with a very good looking variant. If you're in that sweet spot, you're pretty much your matches are probably gonna be real. But if you're matching with anything outside of that sweet spot, there's gonna be a high false positive rate. And we are experiencing a relatively high false positive rate, not enough to exhaust the community at this point, but it will have issues with scalability. And so there we have a number of strategies that we've outlined and we wish to move forward with, including incorporating the mode of inheritance. Increased utilization of phenotypic data needs to be more systematically addressed. And then having a way to score matches so that busy clinicians can actually say, oh, I need to chase that, or that's not likely going to be real. Because otherwise what happens is that you both are notified of a particular match, but then nobody answers your email. And that's not good either. The next thing we need to do is be able to track successes more systematically than we currently are. And so there are a number of metrics that we want to look at, including how many times there was a contact into the Matchmaker Exchange made, how many matches, how many were actually meaningful, how many got published. Those all make sense. Collecting a number of case studies where you've shown that it works will be important for community engagement. How many times when these two people meet each other, like Dave and Isabelle, is there an ongoing collaboration like they currently have, where they've actually brought in other basic scientists to do studies on their new gene? And then looking at the number of people that are actually using the Matchmaker Exchange and how satisfied are they with this. So there's a lot of work in terms of tracking that we need to address. The next thing, and this is something that I hardly understand, but I was sitting in my office with Ryan Whiskey one day, and he was coming to visit, and he was trying to teach me how many patients do you need in a database to solve all of these rare diseases. And he was blowing my mind. But anyway, so they went ahead and actually, as part of our Human Mutation special issue, modeled this, which I think is fascinating. Now, I can't tell you anything about how they did this, but I can only tell you about the output. And basically that if we're gonna, the bottom line is that if we're gonna solve another 2,000 disease genes, we're gonna need 50,000 to 250,000 cases connected in these databases. So that means we're gonna need a lot more friends, we're gonna need a lot more open sharing, and currently the Matchmaker Exchange probably connects about 28,000 cases. So we still have a significantly a significant amount of work that needs to be done, and we need a significant number of sharing. And part of this will actually be about going into the clinical labs and having them share more systematically, because they are, sequencing tens of thousands of patients a year now. The next place that there's been a significant amount of interest is in patient-led matching, and we had a session on this yesterday in our workshop. And there are two databases that are currently connected to the MME that facilitate patients submitting data directly. And right now, patient-submitted data is not being exchanged by the Matchmaker Exchange, but that is something we would like to facilitate, but we have to figure out how to actually make that happen. And so there have been a number of efforts that have been ongoing in this direction, including a patient guideline on how to actually do this, and that is something that we hope to launch in the near future. And then the last thing is expanding the use cases. So currently, as I sort of hope that you picked up, we are talking about two people both having a hypothesis about a patient and a gene. We call that two-sided hypothesis-based matching. But there are other ways to do this because not all data sets have flagged candidate genes or have an idea of what might be in there to discover. And so the first way we need to go with this is the one-sided hypothesis-based matching. So if I have Noah's gene, I can go into other unsolved data sets and manually search that gene for variants associated with cerebellar ataxia in other unsolved patients. There's a lot that's in there for sure that we need to actually be able to expose. And then the other is just sort of having a big cloud in the sky where all the unsolved go up and computers do it for you, which is the ultimate goal. Difficult to do, but there are talks now of a pilot that's about to launch in this area, probably in the next year, for autosomal recessive intellectual disability that we'll be part of. So finally, this is our website. It walks you through all aspects of how to do this. It's recently been updated. It tells people how to get started if you have a case. It tells people how to get started if you have a database. And I direct you to that for more information. And so in closing, this has been work over 3 years of at least 100 people and support from the Global Alliance, as well as the International Rare Diseases Research Consortium and all these other people and agencies and associations along the bottom, because we've all done this, again, with, off the side of our desk. But we have done this because we're committed to giving our patients with rare diseases answers. Thank you.
I often think about the overlap between research and clinical care. And I'm wondering, what is the responsibility of the clinical laboratory to return maybe results that they wouldn't have returned in the past, that are variants that are interesting but have never been associated with causative disease in the past? And I often make this argument because of Matchmaker Exchange that, suppose that's returned to the physician and the physician can find a match. And then the next question is, at what point is it diagnostic? Is it enough just to find one more patient? Do we have to wait until there's a publication? Or how do we make that decision about diagnostics?
And where are you from?
I'm from Cincinnati Children's.
Okay. I think this differs, and we heard this yesterday in our workshop, between where your clinical lab is actually situated. But definitely for those in the US, there are a number of very large labs that report a secondary report, including Baylor, GeneDx, Chicago, the deal, where they actually flag that very compelling candidate gene, whether it's a de novo or a potentially recessive. And I think that I really like that approach, and that is the approach that we'll be adopting in Canada, because it allows for discovery. But once it hits that, it is discovery, thus it is research. And I do think it needs to come back to the medical genetics community to do the things that it needs to do with it to validate that as a novel gene discovery. That's not something that's new to our clinical community. We've been doing that for 30 years since we learned about Down syndrome and things like that. So I think that's an appropriate place for it to go. In terms of validation, it really depends on what level of evidence that you have to say that you have enough to say that you have a new disease gene and that variant is causative. And that still sits on the cusp of translational research. And it's going to depend on how specific that phenotype is and how compelling the story is.
Thank you.
I'm Peter Kapitein from Inspire to Live Amsterdam. I liked your photograph about how it was done 2 years ago. But I needed a bone marrow transplant. And Peter meets a German lady could be the title of your presentation as well, because it was a German lady who had the same HLA type that I have. So I had a match with her. And I think this is awesome because this saves lives. And it's been done before. The database that was needed to make the match between my HLA and the HLA of the German lady, which eventually the one who matched with me, was originated in the 70s of the former century. So it's been done before. That's what I wanted to state. I have not a question. I think this is awesome. This is needed, and we need this for genes, but it's been done before, and I think you can learn from it because the database was originated in the 70s.
I've never actually thought of that, but yeah, absolutely. That's an excellent example.
Yeah, and it was 1 out of 25 million.
We're not the pioneers we thought we were. There we go.
I'm an old-fashioned guy.
Mark Williams, Geisinger. So this is to come back to the prior question about the research and clinical. I think we have to be cautious because we as geneticists are really comfortable with 1 foot in the clinic and 1 foot in research. And we've actually, I think, over the course of 60 years of practice, have demonstrated the virtuous cycle of how moving information from the clinic into the research, working with laboratories, has tremendously improved our ability to do discovery. Absolutely no question. But I think we also have to recognize that genetic testing is moving out of our purview. We're not going to be the only people ordering these tests. More and more people are going to be doing it. And in particular, as we get to sequence level testing that's being applied to populations, perhaps independent of specific indications, returning these types of uncertain results to individuals that are not expecting it and are not plugged in to know how to actually connect up and follow through, I think could really provide a lot of concern. We know from engagement with our clinicians in terms of what they want to get back, they have, not only do they have no interest in this, they are adamantly, violently opposed because they're saying, you're creating a liability, and I hate to use the word for Dr. Knoppers, but
responsibility, yeah.
Responsibility. You're creating a responsibility for me now to follow up with this information that you provided.
Yeah, absolutely.
And we have no mechanism by which we can actually do it. So I think we have to be innovative because we clearly have to share this information. I don't have an obvious solution for that, but we do need to. We can't just dump it on the clinicians and expect that somehow is going to work.
Yeah, I completely get your point. And I think probably I'm coming from too much of a Canadian slant when I talk about, we have more of a system of controlled access to who orders the test than, for example, you would in the US for sure. And the guidelines that we've written for Canadian implementation sort of indicate that you need to have a certain set of skills, and you need to be able to share so we've sort of written the policy and guidelines around that, but I absolutely get your point. And the busy pediatrician is not going to be able to move those things forward, and actually it might be harmful.
Kim, lovely talk. In relation to your red, yellow, and green, how do you sort of do that interactively and make it work in real time? Because obviously it'll be interesting to work out something that's a red as more people put information in it that might start to become a yellow. I was just wondering in terms of
that's a fair point.
How to do that, because I think it would be very interesting to be able to do that over time.
Yeah, so the way it works now is that these matches are made in real time, so they're not stored, right? So they're in real time. So the only way that currently will work with how we are approaching it is if you ask again. So completely get that point, and it's a challenge I think we need to address for sure, Mark.
Thank you very much.
Kim, fantastic presentation. Watching that presentation, something struck me that I haven't thought about before. I want to ask you about it. You say we need to put tens of thousands of cases in these computers so they can whir and crunch and find the new disease genes. And yet your brain, good as it is, sitting over a dinner with alcohol, somehow managed to find matches. Right? Now, you could argue, oh, they're the low-hanging fruit. They're the easy ones.
Yeah.
Right?
Yeah.
But you don't know they're easy until they're solved, if you see what I mean. So did you sit at that dinner long enough to go through 10,000 cases and line them up and find the matches? How did you do it sitting with someone over a table? Can we learn something from that to help our computers do it better, other than pouring wine into the computer, which probably won't work?
Yeah, and so that's an excellent point, Tony. So that those matches over dinner are they start with a phenotypic association. So Bernd works in progeria-like diseases, and Bill, for example, works in overgrowth syndromes, a mosaic overgrowth syndrome. So you just say, oh, hey, by the way, and there you go. So that is, it's driven by sort of the clinical content in those instances, which makes a good point for the big, massive computer in the sky.